Let's look at mosquito diseases. Diseases mosquitoes can transmit include malaria, yellow fever, dengue, filariasis, dog heartworm, and a number of viral encephalitis. In our next slide, the importance of mosquitoes. The importance of mosquitoes may be regarded in three aspects: annoyance, suffering, and death caused by bites and disease transmission is one. Two, economic loss resulting from these diseases and expenditure for medical aid, nursing, medicines, control, loss of time in production. And three, economic loss by holding back development and depressing property value in areas where mosquitoes are plentiful. Let's take a look at mosquito morphology. Mosquitoes are small, two-winged insects in the family Calicoideae and the order Diptera. They can be extinguished from other flies by a long proboscis projecting from the head, the presence of scales on the wing veins, a fringe of scales along the posterior margin of the wing, and characteristic wing venation, the second, fourth, and fifth longitudinal veins being branched. This slide's about mosquito eggs. Mosquitoes lay three different forms of egg depending on their genera. Culex, Culiceta, and Coculatidia lay their eggs in masses of 100 to 300 called rafts. The genus Anopheles also lays eggs in water, but eggs are laid singly on the water surface. Eggs of these groups are laid singly on the water surface and generally hatch in 2 to 3 days. Anopheles lay their eggs in water on the water surface singly, and those eggs hatch in 2 to 3 days. Let's look at the regions. Three regions that mosquitoes have are the head, the thorax, and the abdomen. Looking at the head bears two compound eyes, ocelli or simple eyes lacking. They have long antennae and a proboscis. Antennae are segmented with a whorl of hairs at each joint. The males has dense, bushy hairs, while the female has short, sparse hairs. The female uses her antenna to locate hosts for a blood meal and to find habitats to lay eggs. The male antennae are used to locate females in order to mate. Looking at the mouth parts, the maxillary pulpi sensory mouth parts also differ between males and females. Females have a more slender pulpi than males. The proboscis is a piercing and sucking mouth part and is used by females of many mosquito species to obtain a blood meal. It consists of 6 elongated parts enclosed in a flexible sheath called the labium. The labium is a hollow, cylindrical tube, nearly open along its dorsal surface and terminating in a long pair of lobes called. This slide's about the thorax and abdomen in adult mosquitoes. The thorax is a wedge shape, broader above than below, and bears 3 pairs of legs, 1 pair of wings, and 1 pair of halters or modified wings. The thorax is divided into 3 distinct regions: the prothorax, the metathorax, and the metathorax. The wings are on the mesothorax. So the 3 distinct regions are the prothorax, the mesothorax, and the metathorax. The wings are located on the mesothorax. The abdomen is comprised of 10 segments. The last 2 segments are modified oviposition egg-laying areas in the female, and these areas, the last 2 segments in the male, are for copulation with the female. Let's take a look at the larvae of mosquitoes. The larvae has the same distinct body regions as the adult, although the features are different. The head will bear 2 compound eyes or small accessory eyes, ocelli, immediately behind the compound eyes. They have antennae and mouth parts, which consists of the labrum, mouth brushes, mandibles, maxillae with pulpi, and labrum. Looking at the thorax of the larvae of mosquitoes, it consists of three fused segments. They are not clearly differentiated and can be distinguished only by group hairs called setae found on each segment. In this slide, we're looking at the larvae of mosquitoes. Again, you can see the abdomen is comprised of 9 segments. The eighth segment bears the breathing tube, which may be different in form among many different genera. Looking next in this slide, we're going to look at the mosquito pupae. The pupa is a comma-shaped and divided by 2 regions, the cephalothorax, enlarged anterior part, and the abdomen. The cephalothorax, composed of the flattened head and thorax. On the anterior end of the thorax, there are 2 horns known as respiratory trumpets. These are the external openings of the pupal respiratory system. Continuing with pupae, the abdomen, made up of 9 somewhat flattened, freely moving segments, with the terminal one being smaller than the others. A pair of large flattened plates on the end of the abdomen enables the pupa to swim. Looking at mosquito eggs continued, the genera Aedes, Ochurlotatus, and Saurophora do not lay their eggs in water. Depending on the species, eggs may be laid in damp soil or leaf litter in a woodland depression, or among the margins of a body of water like a pool, swamp, or tree hole. The eggs of these groups are desiccant resistant, and some even require drying out fully before they can hatch. The mosquitoes are known as floodwater mosquitoes because they are stimulated to hatch when there is an insufficient influx of water. Mosquito life cycles. All mosquitoes undergo complete metamorphosis. This life stage includes eggs, larva, pupa, and adult stages. Larval and pupal forms require stagnant water to complete development. Looking further into mosquito eggs, eggs are laid directly in the water or on a solid substrate depending on the life cycle strategy of the species. Generally, if a species overwinters in the egg stage, it lays eggs into the depressions that will be flooded or in the margins of the body of water. So generally, if the species overwinters in the egg stage, it lays eggs in the depression that will be flooded or in the margins of body of water. Okay, so remember that if the egg stage is going to be overwintering, they're going to be in the margins of flooded bodies of water. Eggs will hatch into larvae, which will go through the 4 stages of development or instars. So remember, eggs hatch into larvae, and there's 4 stages of instars. The larvae of most species filter feed on organic matter in the water. So the larvae of mosquito species filter feed on the organic matter in water. All larvae must come to the surface of the water for oxygen. Looking further into the larvae, often called wigglers because of their mode of locomotion. They swim by bending their entire body, causing them to wiggle and twitch. The non-feeding pupae are formed after the 4th instar. Pupae are often referred to as tumblers because they move in a somersaulting position throughout the water. They only remain in that stage for a few days. Let's look at adult mosquitoes mating. The adult stage is when most mosquitoes disperse and mate. Some species fly only 1/4 to 1/2 mile, while others travel up to 40 miles. The average flight is 5 to 10 miles for adult mosquitoes. Adult mosquitoes feed on nectar and plant juices for sustenance. In most species, the females require extra protein to develop eggs, which they do acquire through blood meals. Looking at adult mosquito, adults generally survive for about a week, while females can live up to 4 weeks. Adults tend to favor dense foliage as resting areas. Females are most active at dark, and CO2 is going to be the main attractant to females. Looking at multivoltine types that overwinter as eggs. Aedes vexans, this group breeds in a range of temporary freshwater habitats, and eggs are laid on depressions that flood and dry intermittently. O.C. sollicitans, this group breeds in salt marshes. Females lay eggs on the marsh substrate above the daily floodline. Eggs hatch when the floodline water fills the marsh. This means broods can hatch as often as every 2 weeks in the summer months. They can also hatch after heavy rain or flooding storms. Looking further into multivoltine mosquitoes that overwinter as eggs, the O.C. triceratops, females lay eggs just above the waterline in containers such as tree holds, plant axils, rock pools, and artificial habitats such as tires. The Encephalitis walkeri, females lay their eggs directly in the water of swamp habitats. They lay a different type of egg for the generation that will overwinter. This egg is hardier than eggs laid in the summer. So they have a whole different type of egg that they lay in the overwintering stage on the Encephalitis walkeri. Overwintering in the edge stage allows this species to emerge earlier in the spring than other Encephalitis species which overwinter as adults. They can have multiple generations in a season. Looking at univoltine, that mosquitoes overwintering as eggs, the Otrolortatus stimulans, females lay eggs in damp leaves in woodland depressions. Eggs hatch in the spring when these fill with water. The O.C. canadensis, this group breeds in both transient and permanent habitats. Females lay eggs in leaf litter on vegetation. The bulk emerges in the spring. In the O.C. aberratus, they breed permanent in habitats such as swamps and bogs. Eggs are laid in a waterline, usually on vegetation, and are washed into surrounding habitats with spring flooding. Looking at univoltine as larva mosquitoes, Coquilitidia perturbans is the only species of this type. It's a unique species because the larval and pupal stages breathe aquatic plant roots and do not come to the surface for oxygen. It overwinters in any larval instar stage, so even though it has one generation per year, adults emerge throughout the summer because of the staggered development. Eggs are laid on the rafts of water. Multivoltine mosquitoes as a mated female that overwinter as a mated female are Culiseta melanura. This type lays eggs directly in the water of swamp habitats. It generally overwinters as a fourth instar larva. The larvae burrow into settlements as they overwinter. In the species Orthodomya signifera, breed both in natural and artificial containers. Eggs are laid directly into the water, and these species breed continuously throughout the summer. The larvae need to burrow in the winter to avoid freezing, laying in a tree hole, but maybe they would survive in an artificial container, but they may survive in a tree hole, but they definitely would not survive in an artificial container. Looking at multivoltine as larvae, the Wyomia smithi, the only species of this type, breeds in the water of the pitcher plant. Looking at multivoltine types as mated females, the Anopheles quadricultus, when temperatures are warm enough, the overwintering females emerge from hibernation to lay eggs. These species oviposit directly into pristine fresh water. They peak in mid to late summer. Looking at multivoltine types as mated females, the Culex selenarius, species in this type oviposit directly into the water and can generally breed in brackish habitats. They can breed in salt marshes or freshwater, but they are most abundant in pools adjacent to salt marshes, which are mildly brackish. In Culex pipiens, females oviposit onto the water habitats with high organic contents. They thrive in polluted habitats with high organic contents. These areas include catch basins, sewage, treatment plants, landfills, and such. They are highly attracted to putrid water and are the target species of grave traps. Looking at surveying and monitoring, to determine the number of mosquitoes in a given habitat meets the threshold for larvaciding, use a dipper to collect larvae as they rest under the surface film. Dips should be taken from a variety of locations within each site. By skimming the dipper among the vegetation floating at the water surface, you can collect Anopheles larvae. Looking further into surveying and monitoring the Saurophora and Aedes, larvae are easily frightened and must be collected by plunging the dipper into the water quickly before they have the opportunity to dive to the bottom of the pool. So in the Saurophora and Aedes, the larvae are very easily frightened and you must collect them by plunging the dipper into the water quickly before they get to the bottom. In the Coquiletidia, larvae can't be collected by dipping because they attach themselves to plant roots and therefore you must uproot vegetation to collect them. Upon returning from the field, identify larvae species using a dissecting microscope and dichotomous keys. Larvae can be arranged and reared into mosquito adults and then identified to species. Looking at monitoring of mosquitoes, monitoring can serve several purposes. Trapping can show if the mosquito population is large enough to be a nuisance or if potential disease vectors are present. Changes in the abundance can be used to time adult deciding with population peaks or to evaluate prior control measures. Looking at light traps are the most common method to collect adult mosquitoes. Traps use white light and dry ice as bait. Traps should be hung 3 to 5 feet from the ground and run for 18 to 24 hours. Light traps catch a variety of mosquitoes and represent a large portion of the overall population. So again, traps they use white light and dry ice as bait. Traps have to be hung 3 to 5 feet from the ground, run 18 to 24 hours. Looking at collected adults, when adults are collected, they can be knocked down in a freezer. This preserves them until they are identified. When doing identifications, remove debris, non-mosquitoes, and males from the trap catches so you leave only female mosquitoes. Identify the mosquitoes as species using dissecting microscopes and dichotomous keys. Species should be pooled by species, date, location, and sent to the lab in Concord. You can determine optimal spray dates with these results. Looking at gravid traps and resting boxes, which is another type of trap, the gravid trap uses a hay infusion to attract females that have taken a blood meal and are looking for a place to lay their eggs. These traps as well should run for 18 to 24 hours. Resting boxes are used specifically for Chelicida malinura. Resting boxes are laid in a line on the forest floor. It is best to collect specimens between 10 a.m. and 2 p.m. There are 3 types of mosquito control. The 3 types are biological, cultural, and chemical. Looking at biological control, there are numerous predators and parasites that can affect mosquitoes. Fish, birds, bats, and insects that feed on larvae are examples of this type of control, but they will provide limited results. An example of biological control that uses a pesticide, BTI, Bacillus thuringiensis israelensis. The popular larvicide is applied directly to mosquito habitats where the bacteria kill larvae upon ingestion. So again, BTI is applied directly to mosquito habitats where the bacteria will kill the larvae upon ingestion. BTI provide more control than other agents because the bacteria target the entire population in the habitat. Viruses include fungi, bacteria, nematodes are all considered biological control. Fungi, bacteria, nematodes, all biological control. BTI, biological control. Our next control method will be cultural. All mosquitoes need water to complete their development. Eggs will not hatch and larvae and pupa cannot live without water. Source reduction can include eliminating artificial containers. Ditching, adding ditches to increase drainage is another source. Filling, if you fill low areas and depressions, they can help eliminate breeding areas. You also can look at clearing streams and culverts by unclogging debris. Look at clearing vegetation, removing shelter from predators provided by plant growth, and modifying the habitat. Mosquitoes breed in shallow protected areas. Things like steepening the bank of ponds or dike water flow and flooding the salt marshes can help as well. Looking at chemical control. Chemical control is used where other forms are impractical or along with biological and cultural control. Adulticiding includes non-thermal fogging with products such as etofenprox, which is Zenivex, malathion and piperonolbutoxide, fifanin, and nalid, which is dibrom, permethrin and piperonolbutoxide, biomist, pralethrin, sumethrin, and pipalbutoxide is duet, and sumethrin and pipalbutoxide is anvil. Something else we look at is mist spraying. This would include products such as bifenthrin, deltamethrin, malathion, permethrin, rosemary oil, geraniol, and peppermint oil. Or it could include tal, which is fluvalinate. Looking at larvicides, you could have products such as abate, which is Timphos, Altocid, which is methoprene, Bacillus cehoericus, Vectolex, BTI, which is Vectobac, Technar, Bactimos, and Aquabac, mosquito larvicide oil, BA and Cocoa Bear, and Spinosad, which is called Natular. Looking further into larviciding, larviciding must be well timed to achieve effective control. On salt marshes, larviciding is timed with the tides. Flooding tides occur during a full or new moon. Filling the marshes to high levels and washing mosquito laid in the upper marshes into the water. So the best time to do larviciding is salt marshes, is timing them with the tides. You need to look for a full or new moon because that's when flooding tides occur, and that pushes the water up into the marshes that helps the eggs hatch. Looking at the larvae develop quickly on the marshes in summer. Larviciding should commence as soon as high enough numbers of larvae have been found. This means that the larval stage only lasts 4 to 5 days. Since larvicides require larvae to consume them, there may only be a 4-day window for treatment with larvae, very important. Larvae stop eating during their 4th instar, so stomach poisons are not effective at this time. Looking further into larvae, in freshwater, larval development begins in the early spring after snow melts. Throughout summer, heavy rains can trigger further generations to hatch. Looking at diseases, encephalitis is a disease affecting the central nervous system. It is only transmitted by mosquitoes. Sparrows and red-winged blackbirds are the ones that carry the disease. West Nile virus is also a very important disease. It was first detected in the U.S. in 1999. It is carried by the American robin. Eliminating containers around houses tremendously reduces sources around homes. Heartworm disease is also called by the filarial worm that lives in the heart of dogs. These worms are transmitted by mosquitoes. We need to do an introduction to black flies. Black flies are small, 1 to 5 millimeter humpback insects in the order Diptera and the family Simulidae. They are blood-sucking insects abundant in the spring and early summer. The mouthparts of females in biting flies are piercing cutting. When females bite, they add an anticoagulant to the blood. Looking at the species in New Hampshire, there are approximately 40 species of black flies in New Hampshire, of which only 4 to 5 bite humans regularly. Other species feed on birds and small mammals. Looking at black fly morphology and life cycle, eggs of black flies are laid in the clear flowing streams that are free of pollutants. So black fly eggs are laid in clear flowing streams that are free of pollutants. Depending on the species, females deposit 150 to 500 eggs in masses on vegetation, rocks, and other solid surfaces in or at the water edge, or scatter singly over the water surface where they settle. The smooth, triangular eggs are creamy white when first laid, but become dark before hatching. The eggs hatch within 3 to 7 days in multivoltine species. Looking at black fly larvae, larvae are always found in running water where they filter debris, bacteria, protozoa, algae, and small crustaceans. They anchor themselves to rock or grass in streams by circle of caudal hooks on a button of silk and use cephalic fans to create a current to draw food into their mouth. So black fly larvae attach themselves in circles by caudal hooks, or they actually anchor themselves to rocks or grass by circles of caudal hooks on a button of silk, and they use cephalic fans to create a current to draw food in their mouth. This is how they feed themselves. The larval development period can be 15 days to 6 months in black fly larvae. There are 6 to 8 larval instars depending on the species of black fly larva. When looking at black fly pupa, the pupate of black fly constructs a cocoon of silk in the shape of a cone or loosely fitted sleeve, which is attached to a rock, grass, or debris in the stream. Pupa can be identified to species by the number of respiratory filaments located on the interior of the dorsal aspect of the thorax. Looking at black fly adults, adults are small, stout-bodied, and variable in color. Species in color may range from gray, black, brown, to yellow. The humpback shape of the adult flies is a result of the prescutum and a strong development of the scutum. The eyes of females are more separated than the eyes of males. Ocelli are present, the antennae between the eyes, and have 9 to 12 segments. Looking at adult black flies, adult females feed during the day, with biting peaks in the late morning and afternoon and early evening before sunset. Not all species of adult black flies are blood-sucking. Some black flies have autogenous adult stages where adults do not feed on blood. Most of the life cycle is spent in the egg or larval stage. The adults only live for a few days. Autogeny occurs in the blood-sucking species as well. Males and females feed on nectar, and most females require blood meal for ovarian development. Looking at black fly control in New Hampshire, efforts have actually been minimally effective. BTI has been effective as a larvicide, and little or no effect was observed on non-target organisms. Effective black fly control involves controlling larvae. Local but temporary control of black flies may be achieved by using adulticides such as permethrin, simethrin, or ethofenprox.